So thank you everyone for coming back to the last session of this symposium. And so here we're gonna give you a little bit hands-on exercise and hopefully will enjoy. And so first of all, I would like to introduce the hands-on session teachers, and they have been teaching my class. They're terrific instructors in teaching single-cell biology and spatial biology data analysis. And so it turns out they are sitting in the same conference room. I think my very talented PhD students, Shojun Bao and Alev Baisoy. And I don't have to spend too much time introducing them. I think both are very talented working on kind of cancer biology using a variety of spatial omics technologies. And Shojun, her own research interest in human brain tumor glioblastoma, and Alev is looking into a variety of cancers, but specifically in the context of cancer immune interaction. So they have a lot of experience in cancer biology as well as the techniques used by used for single cell and spatial omics analysis. I'm gonna now hand over to my talented students, Shojun and Alev. You can go ahead and take it away.
Excellent. Hi, everybody. It's so great to be here. And thank you for having us here to lead this, hopefully, really exciting hands-on session. So what we're going to do is start off with a little bit of a background, specifically focusing a little bit more on dbit-seq, since that's what the hands-on tutorial will be involving. And then we'll jump into that hands-on tutorial, which you should have the details for in the chats. So to start off with some of the common spatial technologies, a lot of this has already been echoed throughout the presentations we've heard from today already. So on the left side, you can see the sequence-based spatial technologies, which commonly utilize capture probes or DNA oligonucleotides that terminate with a poly-T sequence that matches the poly-A sequence on cellular mRNAs. And these capture probes are built by means of microarray printing in the case of ST and 10x Visium. And then in SlideSeq and HDST, we see that the probes are synthesized on gel beads, which are randomly attached to the slide, or they can be deposited on a microwell chip as well. And then in SeqScope and PixelSeq, there's the use of gel beads, and the capture probes are produced by modifying sequencing clusters produced by bridge amplification on immune flow cells or hydrogels. But then in the StereoSeq example, they're produced by RCA methods. And then at the bottom, you can see in dbit-seq, the barcodes are created by directly placing microfluidic channels on top of the tissues, so that we create a sort of indexed grid of biomolecules, where we can extract the barcoded information from that. And then on the right side here, you can see some of the image-based technologies and proteomics methods, which can really be divided into two different types of methods here, where on the left, antibodies are kind of bound together and detected with mass spectrometry or codex imaging. And in IMC and MIBI specifically, the antibody is bound to a different metal and is quantified with mass spectrometry. And here the distribution of the metals can be used to deduce spatial distribution of the proteins. And then in fluorescent-based methods like codex, because all the antibodies are bound to the sample at once, the detection is performed in cycles by secondary fluorescent probes, and imaging is pretty quick, but requires custom modification of the antibodies. And then on the right-hand side, you can see the primary antibodies are bound themselves and are stripped after each round of detection or chemical cleavage in the technologies that we see here on the right side as well. So these spatial methods are extremely successful, mainly because they're very well commercialized and available to people to use, and they're really exciting. So to jump into dbit-seq, we have many broad applications of different modalities, which are shown in this slide right here. So on the left, you can see some of the different modalities that we're able to use with dbit-seq, such as histone modification, accessible chromatin, mRNA profiling, and proteins, in addition to many more. And using our technology, which I'll speak about a little bit more in the next slides. And we are able to produce this really great data that we'll be able to take a look at today. Here are some of the publications highlighting the multimodal use of this technology. And I'll jump into the experimental workflow of dbit just as a quick background. So we have our first step where you fix the tissue, fix and permeabilize your tissue, and then you perform bulk reverse transcription. And then you place one chip with 50 different channels on top of the tissue. So this can be 10 micron, 25 or 50 micron. And we place this on top of the tissue and flow 50 barcodes in a horizontal direction across the tissue. And then we do the same thing, but with a second chip that flows perpendicular to the first direction that was flown. So then you can imagine that we create this sort of 50 by 50 grid on our region of interest on the tissue. And then in the next steps here, we are able to perform template switching with our cDNA and then prepare libraries, which are then sequenced. So in this slide here, it's really just to show kind of the actual chemistry that takes place for what I just explained. So here you can see the tissue section on the slide where we can do the fixing and reverse transcription I mentioned, TN5 transposition in this case, and then we flow our barcodes, so showing the kind of multimodal use of our technology. And for example, here with the RNA chemistry layout, you can kind of see how from the poly-T oligo that we use to capture the mRNA, we're able to kind of build out this entire information that we need to pull out the spatial information from each of the mRNAs or whichever biomolecule of interest there is. So we have our barcodes A and B, we have our PCR handle, our unique molecular identifier, and then the biomolecule of interest, of course, and attach the P5 and P7 handles for sequencing. So this is kind of a pictorial just to kind of see what the devices actually look like. So here we clamp on the microfluidic chip onto the tissue, which is kind of in this central area. You can see this is chip 2 flowing in the vertical direction. And then I have a video here, which kind of shows if we have a vacuum on one side of the tissue, we're able to actually pull the barcodes through. So you can see in this video that the barcodes are getting pulled through the tissue, and this is how we are able to create that grid and extract the spatial information. And then finally, here's another image showing, in this case, is a liver tissue prior to barcoding and reverse transcription. And then this is putting our first direction chip on the tissue, our second direction, and you can see it creates, sorry, this is a little bit off center, I think, of formatting, but you can see right here, it should be a nice square ROI for our downstream analysis.
All right, so after we know about the experiment flow for how dbit is attaching the first and second barcode, we want to process our spatial omics data, right? So here is a brief introduction from sequencing-based methods. So originally, when we do the sequencing-based transcriptomics, we do single-cell data analysis. We do single-cell experiments. And the major difference that we have from the spatial data to the single-cell data is kind of minor if you look at the oligo design to capture those RNAs. So as we can see in the upper here is that 10x workflow having the single cell capturing, and they got the cell barcodes in the green region here. So in the spatial omics sequencing based, we just replace those cell barcodes into the spatial barcodes that contains the spatial information. So the most interesting thing we first need to do is extract the spatial information. So here, as we have described it before, that we have two perpendicular chemicals flowing through the microfluidic device. We have our position just as X and Y coordinates. To extract that information, we can either do it manually, or we can use a software that's developed by Atlas Omics. So I will briefly introduce both. I will jump into the GitHub link that's shared in the chat previously. So just to mention again that all the scripts and demo data that we are gonna use today is included in the Google Drive link, and all the instructions is in the GitHub. So if we go to the GitHub page, the first section is about the spatial folder generation. And if you want to install the Atlas Omics browser, it's compatible with Intel chip like operation system. And I will show you how to use that later. And for the manual selection, if you want to do some larger region of interest with your own like customized spatial barcodes, then the software might not be compatible with you. But this is the manual selection method, step by step within the folder here. Actually tells you how to select the tissue like spatial information, whether this pixel is on tissue or off tissue, and the generating the spatial information folder for you. So if people want to do it like step by step, you can just click into the folder, and there is a screenshot and everything very detailed. So I want to show here about Atlas Omics here. Okay. So if you have successfully installed the software, sorry for the delay. So if you really want to, if you have installed the Atlas browser folder, so the only thing you need to do is that start initiated. Oh, sorry. So first you need to contact activate the environment that you installed the Atlas Omics. And then you Python activate this software. As we can see here, that this is the basic software that we are using. And we can actually using the file to begin an imaging process here. So here are several options that we can get. Here is the imaging scan. So I'm just selecting one demo data that's in the Google Drive folder. It's within the 01 point demo data, and it's marked as the Atlas spatial folder generation, and you got the sample information inside. So here, there's a default barcode file loaded here. If you go into the Atlas browser software package, in their folder, there are actually several versions of barcode file that you can select from. You can also customize it. So here you can leave the run ID empty, but I will just put it here, like this is the GBM sample. So you can make your run ID here to identify from your different practice. Once you submit it, it will read the data and it will jump. So here we can see this is a wide field and a fluorescent imaging overlay together to give us the overall region of interest that our barcode A and B flowing through. So Based on your tissue scan, sometimes you might flip it while you're loading the samples. Sometimes you might add it in the wrong way. Like for example, you constantly add your sample on the left inlet and then flowing right one, and then you flip it. You can adjust it at this step. So you can either like rotate it 90 degrees or 45 degrees. This is because there's another design of the chip actually give you X shape rather than the this cross shape for the for your region of interest. So we just confirm our rotation of the original imaging scan, and we can crop this image. So so for this sample, the whole tissue is pretty small. This is 25 micron for each channel. So the region of interest, which marked by the FITSI BSA here, so the green one outlining the region of interest, is actually 2.5 milliliter by 2.5 milliliter. So this sample is pretty small. So if you are getting very big tissue and you are running experiments and do the whole scan over the tissue, it might be too big for you to select the tissue, select the spot, whether it's on tissue or not. So you can choose to crop it. So you can activate it and you can confirm it. So by default, it's going to have this crop image size like this, but you can always change the size of it. You can always move. That will not affect your final generation of the spatial folder. So this is a good part about this software. So once you confirm your cropping, it's pretty slow. Yeah, and you can change the well, this is sharing correctly. All right, so you can change the threshold that whether you want to keep it like as tissue or not. So for this tissue, this is a fresh frozen tissue, and it's a glioblastoma tissue, as Ron first mentioned. Then this tissue is kind of having a lot of debris because the section of this kind of tissue is really hard, and this is fresh frozen. So you got a lot of areas that has some like tissue debris, but this is majorly empty. So you can change the block size and the substrate to change the contrast to find the best contrast about your tissue. And once you confirm it, and you can locate in the ROI. So by this, you need to find each location. So sometimes when you do the experiment, it's not exactly the square. It might, like when you do the experiment, it might tilt a little bit. So that's why we want to aim to the 4 cross point of this region of interest. So as we can see here, this red line actually outlines our region of interest. And to note that is the FITSI line here is our outline channel. So it's either the first channel or the 5th channel, both first direction and the second direction. So after we confirm it, and we can, so now I only have just one TIFF file loaded into it. It has 3 layers of information. So wide field, FITSI channel, and also I did the DAPI staining, which is in the RF, like red fluorescence channel. You can do the overlay with the different black, white field. You can do this like this, or you can put it onto your post-B figure, or you can do it on your BSA. So this squares actually knows the size of the location, the overlay of channel A and B. So each square actually indicates that. So this is easier for our eyes to tell whether this is empty region or this is a tissue region. So we can choose this one. And then we can activate the on and off tissue. By doing that, so because my tissue here looks a little bit like dirty-ish, so it's recognized everywhere has a tissue. So we can manually extract those region like this. So if your tissue, or if while you're doing adaptive thresholding, you set the parameters properly, it should be more neat. Like it will automatically detect those background region for you. So I'm just clicking off several of the dots. So you can see that with this software, you can easily see that the red one indicates there's tissue underneath, and the empty one means that there's no tissue. So I would not just unselect all of the place without tissue, but then we can go ahead and create the spatial folder. So the spatial folder will be generated exactly the same within the same folder that you originally store your scan. So remember, we import the data from the file folder, and then the spatial folder will actually create it within that folder. So once we see this notification, our step with this software is done. And if you want to do more, you can have a new instance here. And it's giving you another one. So you just repeat all the steps for your all samples. So this is a part for software selection of the spatial information. So let me go back to the slides. So all the manual selection process is stated in the GitHub, and we are using Adobe Illustrator and Python. So there's a Jupyter notebook. So the thing within the spatial folder is the JSON file that used to configuration, and also the barcode information that originally is compatible with our experiments. So if you're doing the experiments with your customized spatial barcodes, you should replace that file with your own barcode files. The reason why we want to do the manual selection sometimes is that if you have a bigger region of interest, like 100 by 100 pixels, or if you're doing like using the like your platform is not compatible with the software, then do not worry, because you always have a backup option. That's the authentic way that we originally do the spatial information extraction. So after we extract the information that is compatible with all the downstream analysis, we want to break down our raw data from the sequencing to see how do we get the spatial information out of the sequencing, right? So all of our library is built with Illumina Next Error Kit, and it's sequencing by Nova 600 sequencer. So we do pair and 150 base pair in sequencing. And we can see here from the raw data, this read 1 actually contains the gene or DNA fragments information. And then read 2, as a lab previous introduced, it has the barcode information and the UMI information. Just have the RNA and ATAC as an example here. If you are doing SciSeq, so there will be ADT information here. And if you are doing cut and tag, it will also have the ME region, which is the guidance DNA here at the end of your sequencing. So something I did not plot it here is that after the M region or UMI region and the poly-T adapter region, the remaining base pairs are all biological informations too. So if you're doing ATAC data, then I think the data analysis requests 3 reads. So 1 read contains the spatial information, 2 reads contains the biological information. So the remaining base pairs is actually the read 3 that we need to reformat. So speaking about the data preprocessing, currently the most prevalent ways that we are doing in the lab is SC pipeline and cell render. So SC pipeline is the I think the very first one of the very first pipeline to process the spatial transcriptomics data, and it performs the quality filter and performs the spatial barcode extraction and UMI extraction to find the gene expression matrix in the end. So Cell Ranger has the same function, and also it can process the ATAC data, like it can map the DNA fragments into the whole genome. Both pipelines use STAR, so this is the genome alignment tool. So for people who are not familiar with this part, you can refer to the STAR menu to see how to build your own genome alignment or own genome reference. So in our lab, to keep consistent, we keep using SC pipeline for our spatial RNA-seq data, and for Cell Ranger, we are using it to process our spatial ATAC data. So for those who are experts in Cell Ranger, because I know it's a commercial software, you can always use Cell Ranger to produce your gene expression matrix too. I think it's basically the same idea about extract the spatial information and find the UMI, and also use those information to filter, to map the gene information to the genome and generate the gene expression matrix. So for these two data preprocessing methods, we are also having that in our GitHub introduced here. So if you're interested to build your own SC pipeline processing environment, it's actually built under the Conda environment. So we are using the Conda environment and bash commands to build the environment. So if you follow the step-by-step instruction here, you will finally find the software installed. And once it's installed, you still need to build your own STAR reference and reformat the raw data. As the previous slide introduces, the raw data directly from the sequencer, it makes the biological information and the spatial barcodes and the UMI all together. We need to reformat that, extract that information to put into ST pipeline for the software to properly process and generate the gene expression matrix for us. And speaking of the Cell Ranger, if you want to install it, here are all the dependencies, because it's so Cell Ranger itself is a commercial software already. So you can refer to the user manual there. And after you have already installed Cell Ranger, something we need to customize is the barcode information that's unique for your experiment. So for each experiment, if you have changed your spatial barcodes, you definitely need to replace the barcode information file within the software. So here are the step-by-step and also the way to install the mouse genome and the human genome for the Cell Ranger reference. And I have put everything scripts in the GitHub. And so if anyone wants to try to build the Cell Ranger pipeline, you can refer to the folder underneath the spatial ATAC. So jumping back to our slides, for the downstream analysis, after we have our expression matrix of RNA, whether we are doing multi-ohm or not, we first want to analyze our data separately, because each modality already provides tons of information. That's the fantasy of the spatial omics, right? So the major tool we're using is actually Array. So I'm putting a screenshot of their package here. So as previously mentioned, the major adaptation from the single cell technology to the spatial technology on the sequencing-based track is that we are replacing the single cell barcode to the spatial barcode. So we obviously see the similarity between these two kinds of data, right? So we can definitely apply most of the single cell data analysis package to analyze our spatial data. But there's something I want to note that. So within our spatial data, there's a physical context in the data. So that allows us the potential to do localized analysis. For example, if you're specifically interested in certain region and with the pathological or histological annotation, you already know the functional area about your tissue, about a certain structure, you can look into that. You can select certain data, just like you're selecting certain cell type from your single cell data and do subgroup analysis. And also, while we are doing single cell data, while we are analyzing our single cell data, we have the pseudo-time analysis. And from the spatial data, thanks to our physical context, we can actually see the trend of pseudo-time or the tissue development trajectory spatially. So that's the different part that single cell data does not have. Also, I want to mention that for all the current spatial technology, either they're sequencing-based or they're imaging-based, we can extract the cell boundary information. We can always extract the cell type information according to the marker, according to the staining, but they're not exact single cell. So to do the analysis such as cell-cell interaction, such as like neighborhood study, we still need to demultiplexing our data. So the common ways to do that is that we're applying the single cell data references or single nucleotide sequencing data and integrate together to find the cell types in each spot. So I will skip the part of doing downstream analysis because there are very mature single cell RNA-seq data analysis. And here I want to introduce the packages that we use to analyze our ATAC data or cotton tag data. So for those, I know, so for those who are not very familiar with cotton tag, is that you are having a proteinase A binding to the specific histone modification site, and you have the TF5 transpose protein that actually can cut the DNA fragments around that site. By doing this, you're actually getting those DNA fragment information around the different histone modification. So it means that these two experiments actually gathering the DNA fragment information in very similar format. The biological insights inferred from these two methods are totally different, but then the way how we preprocess it, how we extract the basic information is pretty similar. So as we can see here, RCR actually has a very comprehensive tutorial. If you go to their vignette, you can see that they're trying to extract the peak information, and they are having the peak calling process, do the chromatin variation, and also other analysis, pseudo-time analysis using the peak data. So this is really reliable tool, and so this is on the R software. So other softwares that we're interested in is that actually give us the possibility to integrate the multi-omics data, right? So all the some of the packages I mentioned before, they already have the capability. For example, Array, which is the classic package, we analyze the single cell RNA data, or the like several other, like here we mentioned the SIGNAC. It's also a very novel package, actually have the capability to analyze the single cell RNA-seq and the single cell ATAC data. So the reason why I'm mentioning all the packages here, either paired or unpaired here, they are all single cell packages. So we need to always recall the principle that our data type is very similar to the single cell data. So we can utilize this package to analyze our data. But then we need to add our spatial information, which we introduced at the very first place that we extract the spatial information out of our tissue to integrate it into our data analysis. So for the hands-on scripts, this part, the demo data again is in the Google Drive. The script is also posted on GitHub. So we are going to run through this. So this is a newly developed package called Spatial Glue. These are like Python-based pipeline that we integrate the multi-omics data. So a lot of packages, because a lot of packages are developed on the R environment, and that requires the version control about R, like R and R packages. So to make everyone easy to try on the scripts to do some hands-on practice, we choose the Python version of it. So the package we use to extract the ATAC information is Snap ATAC2, and the package we are using to extract, actually not extract, but reformat the gene expression matrix into N data is a ScanPy. So those are like Python packages that we will use in the hands-on session. So from the published data here, we can already see here is the human hippocampus. I should put a very obvious sign here. This is a human hippocampus. And this data is actually the co-profiling of both ATAC and RNA-seq on the single slides. So the data, so each spot actually is not adjacent slides, it's exactly the same location. So we share the same information both in ATAC and RNA. So if we are running the data analysis separately, we can find very distinct clusters within different spatial locations, as see here. And we can actually see the red cluster here is the DG region, very distinct from the brain region, human brain region. However, if you look into the clusters, you can see that the ATAC data and the RNA-seq data, they share some similarities, but they are not exact same. That indicates the importance of analyzing the multi-omics data. If we only have single modalities, then the information are kind of captured, but the hidden information underneath it might be lost. So underneath is the UMAP of the unsupervised clustering. The gray dots are the single cell ATAC data that we use the public resources to do the annotation. And we can see that our spatial data perfectly map to the different clusters that identified by the single nucleus ATAC. And here, if we see the single cell RNA data, single nuclear RNA-seq data, our spatial data also perfectly match into different groups of single cell clusters. That gives our cell type annotation reviews. So those information are important for the biological meaning to interpret those either ATAC-seq or RNA-seq. Then if we take a look at the spatial glue here, we can see the spatial glue actually integrate the ATAC-seq and RNA-seq data at the very beginning and use the fragments and peak information and the gene expression matrix to do the unsupervised clustering. We can see that the cluster numbers are like increased, and also the structure of these clusters are more distinct. So pick one marker gene for the DG region, PROX1 here as the spatial profile. We can see the spatial glue really captures the biological structure of the original tissue. So now we are going to run through the scripts. So everything, so I have tested it on the free version of Colab. If you are you're using Colab, you just click on the Google Drive and you just need to open it and it will automatically open in Colab. If it's not open in Colab, you can log into the Colab first and then run it. So it will be a little bit slow with the free version. If you're using a premium version of Colab, it might be quicker. So for all of our scripts, we need to install the package here. If you're also saving the spatial data or demo data in your Google Drive, then you can use the Google Drive mount scripts. If you're running on your local computer with Jupyter Notebook, it is okay to pipe install this package inside the Jupyter Notebook, or you can create a content environment that has those packages installed. Just want to note that the version here for the Snap ATAC 2 is necessary to be 2.4.0 because this is updating package. If you upgrade it into the highest version, then some of the commands might change. So it takes a while to install the package. So for the ATAC data analysis, both SIGNAC and RCR are packages, and that really works out well. But then for multimodality data, Python also have its unique advantage to process it. Especially it can use the deep learning or machine learning models to find the weak correlation between the different modalities. Also, it's like easier to do the version control. So here we are using the Colab scripts to do everything. While we are waiting for this, we can go to the GitHub website and click into different folders and see there are like barcode information here and ST pipeline shell script that you can do Spatial thing. So for the ATAC data, we are using everything like need to be replaced, or the files that we are using, we also put it into GitHub. All right. So if you see some error with the pipe dependency, it's okay as long as this package are successfully installed. It is just because the dependency resolver is incompatible with the Colab environment. And we can run the Google Drive contents and let's skip the access to it. Then we just import the package here. So for the raw data, it's all put in the Google Drive again. So if you made a shortcut into your own Google Drive, or you download it to your local computer, please remember to change the direct in directory or output directory. And we are reading the normalized peak data. So take a look at the data structure. Already has the feature about our ATAC data. That we can run it. So just make sure the Snap ATAC 2 version is 2.24, and then we can load the fragments file out of the cell ranger pipeline and import it into Snap ATAC objective. So this process roughly take 5 minutes. So if you have questions, you can put into the Q&A session and let us know.
It's not clear how to run the drive mount.
It's not clear how to run the drive mount. So if you are running this code trunk, it should have a pop-up window. So if your browser blocks the pop-up window, then you might need to look into the address line to see if there's pop-up window, and it will guide you to the login page of your Google Drive. Once it's connect to that page, you just need to authorize the collab to use your Google Drive info, and it should work for the pathway and everything else. Okay. You omit one. No.
Just download to local.
If you go to the Google Drive. Yeah, this step may take a while because the ATAC data is pretty huge. There's no. Maybe in, direct in, change it, direct in. Collab did not open default. Yeah, if the Collab does not open automatically for you, you can search Collab and log into it first. And from that, open it from, you can copy the script into your Drive folder, and it will open it as Collab. Google Drive mount actually can be used not only in Google Colab, even it's in the Jupyter Notebook. As long as you have the package of the drive, you should be able to use that. Yeah, I have never tried it, but you can see. For people who has the HPC, like high performance clusters, actually you can use the Jupyter Notebook to run it. It takes longer than usual. Structure from the end data. Oh, yes, I can explain the data structure from the end data. So if you take a look at it, so this data set that we are running is actually experiments that performed with 100 barcode A and 100 barcode B, which gives us 10,000 pixels in total. And while you unselect those region that does not have tissue underneath the microfluidic channel, you actually have 9,215 spots. So this is the physical location that we have. And the features here is actually the fragments feature derived, like after we preprocess it, you got the features from the fragments file. And as we can see, the end data structure here, we are having the spatial feature, we are having the assay transform feature, and also the peak feature out of the ATAC. Okay, sorry. Oh, sorry, I did. So if I think it's in the direct folder demo, yes. So if you're having trouble with it, like change the code here from the direct in to direct out to direct in, and run it again. So yeah, the data structure of our ATAC data is actually contains the spatial information and also the peak information. And the end data structure, if you go to the end data introduction page, you can see that it's the matrix. End data has different layer of the matrix. So the major matrix is the observations and the so it's the objects and the variance, and then beyond that is the other like variance, OBSM matrix, and the like variance matrix. So functionalities. So generally speaking, the OBS, like it's the row of the end data, and it indicates either like single cell. So each row indicates one cell, or like each row indicates one spot. And the variance is like if you're having gene expression matrix, each column indicates a gene, and in our peak matrix, each column indicates one peak. And also for the OBSM, it's probably easier to have the end data structure figure there. So if you look at the very original end data structure, so our OBS here is actually the spatial information. Each spot is the OBS, and the variance is like each peak information of our like or peak like gene information is stored in the variance. And then the OBSM have our spatial information, and the OBSP actually have some extra information regarding the OBSM and the OBS. Yeah. So, is there any other tools to visualize the end data? Yes, if you are using ScanPy, it actually can visualize the end data structure. Also, SquidPy, ScanPy, and SquidPy. My code is still running, so if the audience has run through this part, you can put a thumbs up in the chat. Oh, yes. So Wang Yun Choi, if you are getting this error saying the Linda requires certain dependency, it's okay as long as there is a successful message at the very end. This error is due to the incompatible package environment, like the dependency resolver here in the Colab, not due to the script. So you can actually run through the script. We just need the very basic function about these packages. Yeah. So I'm also using the free version of the Colab. So if you take a look at here, you can see the resources you're actually looking at. The RAM here is reaching the 2/3 of the allocated resources.
If anyone has successfully run, maybe put a thumbs up in the chat, it would be useful.
So the collab takes longer time. If you have the countdown environment installed on your computer, it would use more resources with your local computer. All right, so it takes like 6 minutes to like load the data into the structure, and then we can do plot the QC results out of the spatial ATAC data. So you can change this interactive into true. That it gives you the selective tools to put on the graph, and you can actually see each dot's information. So here is a TSS enrichment score and the number of unique fragments plot about our ATAC peak matrix data. So TSS is the transcription starting site. Generally speaking, TSS is considered as open in the most cases because this is a transcription starting site. So this enrichment score actually considered as a very important QC factor. Per cell rangers pipeline, they consider TSS larger than 5 as good quality data. But as we can see here, if we have more unique fragments, sometimes it might get lower average TSS enrichment score. Here is already a normalized data type. And we can see that average of our data actually have more than 10K of unique fragments, and majority of them have the TSS enrichment score larger than 5. So it means that the data of the experiment actually has very good quality to go through. And then we need to add the tile of the matrix. So this step also takes very long. You can run ahead, and then we can take a look at the data structure. So the drive mount.
That was the first question we answered.
Okay.
Mhm.
Okay. Time soon. Okay. So yeah, we are leaving our email address in the chat in case people want to further ask us about questions. Yeah, so we can see now the end data object with the snap ATAC information, like data structure stored here that have the observations as the TSS enrichment score, the number of fragments, and the fragments that have the duplicate, consider has a duplicate, or the fragments that has the mitochondrial gene. So the reason why it's distinguished the fragments with the duplicates is that this package is used to process single cell data. And as we know that for each DNA location, it will have at most 2 fragments. But this is a spatial data. So you know that for each pixel, it might be a mixture of several cells. So it might be possible that the fragments has more than 2 copies. So so if we want to further dig into our data, this might be an important parameter that we look into it. Then we're using the snap ATAC to select the top differentially expressed features while running the model. So we are selecting 250,000 features of our data. Then so below is a function included in the snap ATAC 2 that has the dimension reduction. So for the sequencing data that we are having, it's really, really high dimensional. It contains a lot of peak information. It contains the fragments information, and it has different locations. So we need to do the dimensional reduction just as we process the single cell data. This data set that we have imported already performed the assay transform, normalization and stuff. So we can just run with it. If you're having your raw data, you probably want to first reformat it and pre-process it before you load into the Snap ATAC too. So here we need to refer to the Snap ATAC 2 manual about how the features are selected. Here we can see that I think these features that we selected here is actually the top differentiated features. So that's why if your data does not have that kind of variety, it will not have enough features as you stated here. Yeah. Then it's running the K-neighborhood network and also the latent plot. What do these features mean? So the features of the ATAC data generally means that the peak information that we have out of the cell ranger. So after the cell ranger, we are getting the fragments expression matrix. So for the ATAC experiments, we are getting the DNA fragments around the open region. So if it's openly accessible, then the enzyme will cut those regions. We will collect those region information. And while we are mapping it back using the genome reference, after align it, we're actually having a certain range of the genome has a lot of peaks, a lot of fragments. And those fragments gathering together have certain profile throughout the genome. So at a certain genome location, or we can see the region, we actually have certain peaks. So in certain region, it has higher fragments than the other region. And this region considered to have a peak. And those information considered the feature here. So with those like different features together, we are calling the peak using the Snap ATAC. So just to remember that there's a default co-peak parameter. It's if I remember correctly, is 2 kilo base pairs, 2K base pairs around the center region. So if you are study the specific question that you have different requirements to call the peak, you can change that. Like you can change it to larger region, for example, 5K base pairs, or you can change the narrow region like 1K base pairs. This step also takes a very long time because it's calling the peaks from the 250,000 features that we previously sorted out. Yeah. to save time, I'm running the following steps all at once. So if anyone has successfully run the script, you can always put a sum up in the chat or Q&A. How does the spatial ATAC analysis different from single cell ATAC analysis? That's a very good question. I think the answer to this question is similar to how does spatial RNA analysis different from single cell RNA analysis, is that we first run the QC and the unsupervised clustering, treat our spatial data as single cell data. Even though we know that each pixel has a mixture of different cells, but then each location should have their differentially expressed genes or differentially different open accessibility of the chromatin, right? So even it's a mixture of the cells, then different locations should have different open accessibility. So we first do the ATAC data analysis using the single cell pipeline. And then while we have the unsupervised clustering of those differentially expressed features, we map it back to the spatial location. And with the spatial location, we can do the pseudotime analysis and also the gene rectifier network to see whether with the spatial context, they're showing the trajectory of the organ development or disease progression. We'll talk to that in detail while we are running the spatial group part. So the second part of the script. So as previously mentioned, the spatial information actually, so the importance of the spatial data is that we have the physical context of the original tissue. But then the data structure and the nature of how we capture those information is very similar to single cell. So that's why we always perform single cell like analysis first and run the space map it back to the spatial physical location and see if there's biological meaning with underneath it. Taking longer than 4 minutes, but yeah. So if we go back to see the cluster here, while doing the ATAC data analysis, it's always having the clusters first before we call the peaks. Not like the single cell RNA-seq, we always do we identify our clusters with the differentially expressed genes, right? So here, because the original data from the ATAC after preprocessed by cell ranger is a fragments matrix, and the fragment matrix already have differences. So by this original matrix, we can already have the unsupervised cluster rate that indicates each cluster has different fragments behavior. And based on that, we're calling the peak. Yeah, so this is something different from single cell data. Oh, So the mapping back to the spatial location. So if you recall the first part of this hands-on workshop, we're introducing two different methods, either manual selection or the software to select those tissue location that actually have the barcode flowing through it. And then we are generating the spatial folder containing those information. While we are doing the downstream analysis in the later half of our workshop, we will use the information within that spatial folder and add that into our data structure, into our end data. So I'm recalling the one question before asking about the end data structure. So those spatial data should be in the undefined. So it's in UNS spatial folder should be within that place, because throughout the data structure, it only has one unique spatial information, physical context. And with that information, we can map our map each dots. So here each dot indicates one spatial location. Just similar to the single cell data, each dot indicates single cell. Yeah.
And I can also add that for the RNA, just if you're doing RNA, not considering ATAC, the most basic structure, the output of that ST pipeline that we were talking about is going to have your spatial information in the rows. So like barcode 10 by barcode B11, for example. And then all of the genes will be the columns. So that is the TSV file that's generated. And then that will ultimately become a part of your Seurat object that you're manipulating. So that information is going to be carried through no matter what downstream analysis you do.
Yes. So either you're using R package like R data structure or Python data structure, there's always a location that you can put your spatial information into it. And then with that added into your data structure, we can use those packages to map back the spatial information. So here, after we call the peak for 8 minutes, like 9 minutes, forever, you can see the data structure afterwards. So we're selecting original feature. If you remember, we are selecting 250K. But after we call the peaks, the peak information added to the features. So our number of variants actually increased to 600 to like 6 million features, more than 6 million features. That increase that is because for each peak, so for each spot, it will have a like information throughout the whole genome about each site of the genome has a peak or not. So that is very, very huge dimension of data. And if we take a look at it, like the transcription starting site enrichment score is capped, number of fragments is capped, then we have a latent space that actually doing the unsupervised clustering. Okay. For the it's really weird. We're encountering an error here. Really weird. I'm not changing. Yes, Sarah is answering this as spatial information. It's within the spatial group folder, but also, yeah, it's if you're running the second part of the spatial group, it's actually having the spatial information. So sorry that this having the unexpected error here. This error is probably due to the Python version here is higher than the Python version that's required by the package. It did not appear while we have the dry run. In that case, we cannot plot, like normalize the peak matrix and do the log profile of the peak matrix. But this does not affect our downstream analysis because the output here we saved is also within our Google Drive folder. If anyone in the audience can run through this like successfully, just let us know. When we run through this pipeline, it did not have this error message. Yeah, and then within the script, we're also importing our RNA-seq data to build the end data structure that's compatible for the multi-ohm data analysis. And we can see here, it's also loading the barcode, spatial barcode information to extract the from the expression matrix. As Alav just said, each row indicates the spatial location of, for example, each row has information of x-axis and the y-axis. And if you have that information, you just need to decipher it. You need to split into x and y and put it into the spatial information, like put into the structure with the spatial information. So someone is asking the latent clusters. So latent is actually a model that we are using to do the unsupervised clustering. This is a specifically in the Python, but sometimes I think also see it in the R. So this is just a model that when we run unsupervised clustering, it's calculating the weight of different features and find the distance between each spot and classify it, whether it's close or not. So changing the resolution of the latent structure, like changing the resolution of doing the unsupervised clustering definitely changed the number of the clusters. Then the number of clusters here not necessarily associated with the peak calling results, but if you're having if you're doing so here, it's not necessary change the peak calling results. Yeah. So Sarah said does not work for you. So you're saying the data is not in the folder? Really weird. Let me finish the this code really quick because here RNA data is easier to process. Here we can see the observation names of the RNA data. Here each oligo indicates the spatial barcodes. So each one is 16 base pairs, and that's barcode A plus barcode B, so 8 plus 8. So we decipher this information and make it turn into X and Y. Here, this is because the peak mat was not reproduced due to the error here. We have the data saved already, so we can directly go to the second part. Let's go with the second part then. Let me see. And again, the version control here is very important. If you change the version here, it might needs another update for the requirements, the dependency like version.
Yeah, I think maybe trying to download this locally. I think it just has to do with the input of the data.
Mhm.
So maybe if you could try running it locally and downloading the files, and hopefully that works for you.
Yeah. After we install the package, we just need to import the package. So this data does not work. Oh yeah, so for this, and yeah, we haven't solved this issue. This is probably because the Python version is too high in the collab, because for now we are using 3.10, but for the Snap ATAC 2, it's actually requires the Python version for 3.8. Yeah. So sorry, this part did not work well. While we are waiting for the installation of package and importing the package, we can take a look at the spatial information again. So this like shortly pops up in the last part of the Snap ATAC 2. But if we can take a look here, this 8 region, this is like 16 base pair oligo here. The sequence indicates barcode A and B again. So it's like the first comments indicate that whether this this location is on tissue or not. So it indicated as 1, 2, 3, 4, 5. And so for the fourth row, it's like this is the okay. Oh. I need to restart. Oh, I think this error is probably because I'm running the 2 collab at the same time, and there's a conflicts with the packages. So if you are meeting the same issue about the error, then yeah, just restart the session and run it again, and the package should be imported successfully. And you need to sign into your Google account. Yeah, due to the time restriction, we might not be able to finish everything in the following part, but it should have the like data we need. So every step, if there's an intermediate results, it's all saved in the Google Drive. So you can go ahead and find it in the Google Drive and plot it. So ideally, if we run through the whole script, we'll actually have the spatial plot here to indicate the clustering. So this spatial glue, as I introduced very briefly, it's integrating the RNA and the ATAC data together to have the spatial information. So on the left side is unsupervised clustering based on these two modality of the data. And as people are asking, how do we find the spatial correlation with it? Like each spot, each color indicates a different cluster, and each dot has a spatial information. So we map it back and arrange it as a regional spatial organization, and we can actually see the cluster here. So the clustering 2 here is the DG region that actually shows the PROX1 expression perfectly overlap with this. Yeah, pretty much so let me run through it and see if we can get through it. So for the spatial glue package, you can select the data type. And as we are processing the spatial ATAC and RNA-seq data, you can choose the spatial epigenome and transcriptome type. And it used the machine learning to build the model. Integrate strategy. So for the integration strategy of RNA and the peaks, so I don't know how exactly the spatial group this package does, but generally speaking, for the RNA data, we're having the gene expression info. The mRNA information we extract, it always have the gene information associated with certain mRNA, right? Whether it's a pseudogene or it's like, so for mRNA, it always have the gene information related to it. For each gene, it has a specific location on the DNA, on the chromatin. And then for the peak information, as I have briefly explained a little bit, is that it's the gathered information about the DNA fragments counts within certain region of the DNA. So by nature, they share the information on the chromatin that in certain region, either it's a gene body or it's an enhancer or it's a promoter, the accessibility of this certain region is reflected with the peak. So while integrating the data, they're correlating the peak information with the gene on the chromatin. So it also have the information from the genome, and then they can integrate the data together. And the relatively high gene expression level or low gene expression level from the different location is the features that we are comparing. So as we are integrating the data, the differences between the peaks in each spot, like the peak matrix refer to the open or close of certain region of the chromatin. And then the gene expression high or low. So they kind of integrate those features together and compare each spot, like compare one spot to another, whether these two spots are having similar profile across those two modalities. That's how the package do the integration. For the algorithm, how it's realized this function, actually, I don't know the details, but you can go to the spatial glue tutorial, and they probably have the their methodology introduction there. Before we end it, I will keep running this script. So you can feel free to send us email if you're having any trouble running the scripts here. I want to still emphasize the importance while we are running the spatial data together, integrated together. So if we look at the published data, it's here, here on the upper part here, we are running the spatial ATAC and the spatial RNA unsupervised clustering separately. And this is the mouse embryo data. And we can identify each organs very clearly from the RNA-seq part of data. However, when we do the joint clustering, when we have the multi-omics data integrated together, it actually adds the details into the cluster very differently. Take the cluster J4, like on the third graph as example. It's a cyan color here. And then if you go back to the similar location of the RNA-seq and ATAC data, you can actually see that neither of these single modalities distinctly shows this cluster very, distinctively show these clusters. So if we integrate the two modalities together, we definitely add more details into the clusters. Also, when we do the spatial ATAC and RNA-seq data, and we compare the gene activity score inferred from the ATAC data, or the gene expression score, expression level directly from RNA-seq data, we can see that they have different informations. With those two modality information altogether, we can run the slow time data analysis. So here, sorry. Oops. So for here, the slow time data analysis showing the radioglia to the post-mitotic premature neurons. So the score here, while it's lower, it indicates it's more similar to the expression or chromatin accessibility profile of the radioglia. And if it's getting higher, like yellow color indicate here, it's more like the post-mitotic premature neurons. So from the spatial information, we can actually see the tissue structure, and it's the radioglia layer developing towards the post-mitotic premature neurons. And while we are looking at the slow time analysis here for the RNA and ATAC together with those marker genes related to the embryonic development, we can see that the expression level of the genes sometimes are not linearly correlated with the open accessibility of the certain gene region. So the gene activity scores calculated from the ATAC data indicates that whether this location of the gene is being openly accessible, and also whether this gene has the possibility to be transcribed. Then the expression of the gene is actually the mRNA that we captured at this point. While doing the pseudo time analysis, we can see that sometimes the mRNA has very long lifetime, and even though the chromatin of the chromatin accessibility of this certain gene point is like low, like this is not being transcribed, then the transcription level, the mRNA level is still high. But then while some of the mRNA has very, very short lifetime, then it needs this spot to be open all the time. And this really indicates the gene regulatory network that cannot be inferred with single modality. So we just introduced very brief about performing the multi-omics data analysis here. And there's some errors while we're running the script. Very sorry for that, but we hope this hands-on workshop just provides you different package information and the guidance that you can explore through it. And the environment that we provided in the script and the demo data should give you very fun materials to explore for the multi-omics. Yeah, thank you so much.